So I think to start, it'd be great if you could tell us what inspired you to undertake this study of cannabis and focus on developing a gene expression database.
Okay, well, I would say that the recent changes in the regulation of cannabis use and cultivation around the world have opened up opportunities to research with these species to be conducted now, more broadly. So this has led to a rapid increase in the amount of information available about cannabis. So including RNA sequence data. And however, these data are often in the raw form, meaning that while the RNA-seq sequence files are available in public databases, for example, without proper filtering and analysis, they aren't very informative. So, and additional to this, not all of this data are linked to published scientific studies. So this is where the idea of the Cannabis Expression Atlas came in. The goal was to create a database that translates the RNA-seq data, so to speak, transforming raw sequence information into easy to use and refined data to anyone interested in it.
Great. So you're translating that data to make it more useful.
Yeah.
Excellent. So one thing I noticed when I was reading through the abstract in the paper is that you chose to use the Jamaican lion reference, which again, near and dear to our heart at Medicinal Genomics. Our team is the one who put that together. So how does that reference benefit the study?
During my initial research, I realized that there was no consensus about which reference genome to use in cannabis research. So this made me want to look further to available genomes for this species to choose the most suitable one for use in our study. So I found that most of other genomes came from female plants and were from type 1 or 3 chemotypes. And that's when the Jamaican lion genome caught my attention, because first it's a type 2 variety, so which means that has the both THCAS and CBDAS genes active. And additionally, both male and female plants were sequenced. And the researchers from the Medicinal Genomics had identified a region corresponding to the Y chromosome. And this is a key feature compared to other available genomes, because as I mentioned, are all from female plants. So using this genome as reference allow us to enrich our database and identify expression patterns for Y chromosomes genes, for example, which can be very useful for helping researchers to better understand sex differentiation in cannabis, for example.